Remember, we said last lecture about the process of sensation and how this information will be transmitted through a series of neurons to the central nervous system. Now, how do these series of neurons communicate with each other? It's through regions called synapses. And a synapse is a region where communication between two neurons or between a neuron and an effector cell. And this synapse, the communication through the synapses is not always straightforward or just as simple as one-to-one communication. It can be very complicated, can get very complicated. These signaling pathways or the signals or the action potentials between these neurons can be blocked in the way or can be repeated several times or can be integrated with other types of impulses, and this is what we call the synaptic function of neurons. We're gonna learn more about it later. So, dear Smurfs, here is a type of a synapse between a neuron and another neuron. So it's an area of communication between two neurons. I said the signal may just pass between this neuron to the next neuron, just as simple as this, or it may be blocked and preventing from transmission. Can be repeated several times for doing certain function. So what are the types of synapses? Synapses can be classified into chemical synapses or electrical synapses. In electrical synapses, the cytoplasm between two neurons or two cells are connected by gap junctions, and so the current or the action potential can be transmitted through these low resistance areas between these types of cells. That the transmission between these cells that are communicating through electrical synapses can be bidirectional, from this cell to this cell or from this cell to the next one. This type of synapse is very quick type of transmission of information or action potential, and it's important as forming a unit, which we call it synchronization. It can be occurred in several places in the central nervous system, just similar to the, for example, the cardiac muscle cells, in which they are synapsing, electrical synapsing, forming such a unit of synchronization. A more common type of synapse is called chemical synapse, in which there is a gap, there is a synaptic cleft between two neurons. The first one we call it presynaptic neuron, and the second we call it postsynaptic neuron. As you see here in the chemical synapses, because of the presence of this space between the cells, action potential cannot be transmitted directly. Therefore, another way of communication will be, which is a chemical way of communication, releasing what we call neurotransmitters. Now, the presynaptic neuron, it has large amounts of the synaptic terminal with mitochondria, which is very important to produce energy, important for forming and releasing these neurotransmitters from the synaptic vesicles. And important structure that's present in the presynaptic terminal is the synaptic vesicles. These vesicles, large amounts of vesicles containing different types of neurotransmitters that will transmit this information resulting from the action potential coming here into the next neuron or a postsynaptic neuron. Voltage-gated calcium channels are very important structures located in the presynaptic terminal. Once the action potential comes to this terminal, it will activate voltage-gated calcium channels, resulting in influx of calcium. This influx of calcium will help for exocytosis of these synaptic vesicles, so the release of neurotransmitters. Once neurotransmitters are released from the presynaptic terminals, it has to act on specific receptors that are present on the postsynaptic terminal. And these type of receptors can be either ion channels, so that's what we call it, ionotropic receptors. These ion channels can be either cation channels, positively charged ions, or anion channels for negatively charged ions. For example, and a common example of the cation channels is the sodium channels. So influx of sodium into the postsynaptic terminal, you know that it will result in depolarization or activation or excitation of this postsynaptic neuron. Whereas activation of anion channels such as the chloride channel result in influx of negatively charged ions that will result in hyperpolarization and so inhibitory or inhibition of the postsynaptic neuron. These ionotropic receptors are characterized by quick function, so they are very fast or faster than the other way. Metabotropic receptors. These receptors usually use a second messenger system such as the G protein coupled receptors. These receptors has a slower action than the ionotropic receptor. However, it can be more prolonged action and can be such example for that in memory use. This is showing an example of metabotropic receptor. You don't have to know these biochemical details. An important characteristic of the chemical synapse that it's a unidirectional or one-way conduction at the synapse, which means that the type of information or the transmission of information will be always one way from the presynaptic terminal to the postsynaptic terminal, unlike in the electrical synapses, which were bidirectional. Synaptic delay is another important feature for the chemical synapse. So it's a slower type of communication than electrical synapse. Within hundreds of a second, that's needed from the action potential in this presynaptic terminal until it's formed in the postsynaptic terminal, which includes several steps such as activation of the voltage-gated calcium channel, then the series of action resulting in exocytosis of these synaptic vesicles, the release of a neurotransmitter, then moving through the synaptic cleft until binding to specific sites on the receptors, whether it's ionotropic or metabotropic receptors, and then results in changes in the membrane potential and the formation of action potential in the postsynaptic terminal. All of these steps and changes results in a delayed type of transmission of information in the chemical synapses. So usually there is a relationship between the amount of a neurotransmitter released into the synaptic cleft and the number of calcium ions that entered through the voltage-gated calcium channels. So one of two actions will result in the postsynaptic neurons. Either it will be excitation by opening of sodium channels, decreasing the conduction of the chloride or potassium channels, or changing in the metabolism on the cellular activities, the number of receptors within this postsynaptic neuron. On the other hand, the change can be inhibition of the postsynaptic neuron, resulting from activation of chloride channels or potassium channels, or decrease the activity or changing the metabolic activity within the cells and the number of receptors that are present on the cell membrane. In general, neurotransmitters that activate cation channels are called excitatory neurotransmitters, whereas those activating anion channels results in inhibitory neurotransmitters. And this opening and closure of these ion channels is very quick. So there is a quick action of these transmitters resulting in activation of these ion channels. However, as we said, that some functions of the nervous system, they need more prolonged action than just opening and closure of these ion channels. That's why there is activation of the metabotropic receptors that use second messenger system. So synaptic neurotransmitters are classified into two categories. The first one is called small molecules rapidly acting transmitters, and from their name they are most likely to work on ionotropic receptors, quick action and very rapid. However, the other type is neuropeptides, and these are more prolonged action, mainly working on the metabotropic receptor and changes in the metabolism of these cells. Now, the small molecule rapidly acting transmitters, they are synthesized within the presynaptic terminals. The machinery for their synthesis are present in the presynaptic terminals, and they will be released very quickly when the action potential comes, and most likely they will work on the ion channels, ionotropic receptors, and then they are most likely to be recycled within this presynaptic terminals. They are This is an example of small molecule rapidly acting transmitters. And as we said, the syntheses are present in the presynaptic terminal. This is acetylcholine synthesis, and it will be released quickly from the vesicles, act on the receptor, then it will be recycled again and resynthesize within this presynaptic terminal. Now let's talk about examples of these small molecules rapidly acting neurotransmitter, and one of the most common examples is acetylcholine. Acetylcholine is important to know that it's the only neurotransmitter at neuromuscular junction, which is the synapse between the motor neuron and the skeletal muscle. It's also released from preganglionic neurons of the autonomic nervous system and the postganglionic ones of the parasympathetic system. Most of the actions of the acetylcholine is excitatory effect. Another example is norepinephrine, that's secreted from different neurons, such as the ones in the brainstem and the hypothalamus. Some of actions could be excitatory and others can be inhibitory. Glutamate is almost always an excitatory neurotransmitter that present in different areas of the nervous system, including different areas of the cerebral cortex. Glycine is mainly an inhibitory neurotransmitter that's mainly present in the spinal cord. Dopamine is an important neurotransmitter that's released from areas such as substantia nigra and it's related to several disorders. GABA is always an inhibitory neurotransmitter that's released from different areas of the nervous system. Serotonin is a neurotransmitter that's released from areas such as the median raphe of the brainstem, and it acts as an inhibitory of pain pathways, also plays a role in the control of mood. Nitric oxide is an interesting neurotransmitter, a small molecule that's not pre-stored in vesicles or pre-synthesized. It's only synthesized once needed and diffused through the membrane to the postsynaptic neuron to do certain actions. Now, the other type of neurotransmitters is in neuropeptides. Neuropeptides has a slower type of action because they mainly act on the metabotropic receptors and second messenger pathways. The synthesis of neuropeptides also is not in this presynaptic terminal, but it's in the neuronal cell body or the soma. So it's synthesized by ribosome, then packaged by the Golgi apparatus and endoplasmic reticulum. Because of their complex synthesis, there is a smaller amount or quantities of neuropeptides present in these presynaptic neurons. However, they are more potent than small molecules transmitters. The speed of transmission from the soma of the sensory neurons or the cell bodies to the axon terminals is at slow rate for these neuropeptides, and they are not recycled in the presynaptic terminals, but they will be autolyzed and not reused. The quantities, as we said, they are small but more potent, and the action is more prolonged because it's mainly acts through second messenger system. This is a table showing different types of neuropeptides, and the list is prolonged, but in general, to know the categories, that the hypothalamic hormones and the pituitary hormones, insulin, glucagon, also these are hormones released, enkephalins, which are related to pain pathways. These all are types of neuropeptides. Angiotensin 2 and bradykinin, also considered as neuropeptides. Now, fatigue can take place at the synaptic transmission, and it can be due to several reasons. One of the most important is the exhaustion or the depletion of the neurotransmitter from the vesicles due to continuous activation and release of this neurotransmitter from the vesicles, and there is not enough time for recycling of these transmitters. The other reasons could be that inactivation of receptors in the postsynaptic neurons, or changes in the concentrations of ions in the postsynaptic neurons. All of these changes can result in fatigue of synaptic transmission. Many factors can affect the synaptic transmission, including the changes in pH. For example, alkalosis can increase the neuronal excitability, resulting in seizures. You may have noticed that some people who have panic attacks, when they hyperventilate, breathe very quickly, the CO2 levels will decrease, and that will result in increase in the pH, alkalosis, and that can precipitate these epileptic seizures in those persons. On the other hand, acidosis can depress the neuronal activity. For example, in patients with diabetes or sukari, when they have this diabetic ketoacidosis, that will result in coma or neuronal depression in those patients because of changes in the pH. Normal levels of oxygen is important to maintain neuronal excitability, so any cause of hypoxia decrease the oxygen supply to the brain, can interrupt the function and result in coma or unconsciousness. You know that caffeine in coffee, theophylline in the tea, and theobromine in cocoa can change the neuronal excitability by reducing the threshold for excitation of neurons. Also, some anesthetic medications can increase the threshold for excitation of neurons, resulting in decreasing of synaptic transmission.